Okay, so we're in an alfalfa field here, and this alfalfa has just been raked for baling. And as you can see here, this alfalfa will consist of a mixture of stems and leaves. And we hope that most of these leaves will be retained in the baling process. And oftentimes in the western states, we see farmers baling under dewy conditions in the late night or early morning, and that's in order to retain leaves. So the sampling process has to represent the leaf-stem ratio here, but also we do have a few little weeds in here that are present, and so that has to be represented in the sample. And also we have to see across this field, this is about an 80-acre field here. If you look across this field, we have to represent the soil variation across that field. And the other thing is, and that I'll have to, as a separate issue, point out here, you notice that some of these plants are bleached by the sun. That is, they laid on the windrow at the top of the windrow and got some sun bleaching. We have done some experiments on sun bleaching and found that there's no difference due to sun bleaching in the average forage quality and either the protein or the fiber values, relative feed value, or TDN of alfalfa hay. Bleaching of color does not make a difference, and so people should not look at color as being a primary determinant of forage quality. Although most people like color, most animals don't respond to color. They respond much more to odor and also to forage quality in terms of leafiness and protein and other kinds of factors. Here, this is showing the weeds. Let's see, I'll show you maybe some bleached hay. So here's some bleached alfalfa, and we have non-bleached alfalfa. Now, other than the leaf-stem ratio, which is pretty important, the differences in color shouldn't make a big difference. These weeds, however, may determine somewhat lower quality in this hay. And so this is going to be a medium quality or even a fair quality hay. Okay, so what are the determinants of forage quality? Well, one of them is going to be percent bloom, or the degree of bud formation in the hay. This is mid to late bud and early flowering, or even mid flowering in this field. But the other determinant is actually plant height. So if you take a look at this group of stems, you can see there are some stems that are very, very tall and coarse, and then there are some stems that are very slim and low in fiber. These are very high in fiber. So the sampling process will have to represent the stem differences between some of these earlier growing stems and some of the ones that might have emerged later in the growth of the crop. So the flowering is one indication of quality in the field, and you see this quite a bit of flowering here, and this is in full bud. And so that's one indication, but plant height is another very good indicator of quality. So the taller the crop is, generally speaking, the lower in quality the crop might be. So that's one way to predict quality in the field. Now, in the terms of the sampling procedure, we're going to have to represent some of these plants that have fairly slim and low fiber stems, as well as those that have fairly well developed secondary cell walls, and those are going to be very high in fiber and high in NDF, low in RFV, and low in TDN.